Welcome to this session in pharmacology. Previously we looked at some very important ethical and legal considerations around the use of medicines, whether they are Western pharmacy or complementary medicines. We then looked at how in all of the literature, pharmacopoeia and so on, drugs are listed by their generic name. We talked about the difference between that generic name and brand names, which your clients will more commonly use. We're now going to have a look in some more detail at those key principles that we mentioned, pharmacodynamics and pharmacokinetics. These key principles that we said even apply to the use of complementary medicines. So we saw that the key processes that affect the drug levels in the body once that drug is administered by some route are absorption, distribution, metabolism and excretion. In general, drugs when administered will make their way into a systemic circulation, though we will see that that does depend upon the route of administration. All of those key processes will determine this pharmacodynamic phase, how the drug affects the body. And all of these were how the body affects the drug. And we did talk about a previous phase, the pharmaceutical phase, that is most important in complementary medicine and in Western pharmacy to determine what formulation would be used and therefore by what route it would be administered. So we're going to see how all of those impact on what concentration of that drug actually reaches its target tissue. And remember that those targets were largely proteins. They might be lipoproteins, they might be glycoproteins, but the drug's target is generally largely protein. So two other key terms: a drug that binds to that target, activates that target receptor and produces the response just the same as the endogenous substance does would be called an agonist. So note this term ligand, something that binds. So ligand is just a more general term. Certain types of ligands that bind are the endogenous substance, exogenous substances such as drugs that bind as agonists. When we look at some of the drugs affecting the body's systems, we'll see that clinically it's really important to distinguish those drugs that might be partial agonists. And today we're just going to have a look at, in terms of this key underlying principle of dose of drug, response to drug, we're going to mathematically measure what it means to be a partial agonist. But because it's an agonist, it binds and it produces a response, but we'll see that it's not the maximal response. Another key term will be antagonist. An antagonist also binds, so it's another ligand, but in this case it blocks the receptor. So the endogenous ligand cannot bind, and we'll also see shortly that other exogenous, external agonists might also be prevented from binding, but with that endogenous ligand it's prevented from having its normal response, so there is a diminished response, and we'll look at the mathematics of that. So with these agonists, partial or quite strong agonists, or we will have with antagonists, whether they are competitive or irreversible antagonists. So if they're competitive, they are generally reversible. So stressing that with antagonists, they do not cause the effect, where effect is the normal physiological response. They block that physiological response. They block the interaction between receptor and endogenous substrate. And now we've built up a picture in your mind of that receptor as a protein with active site or a particular shape that binds. And so now we'll be looking at how antagonists affect that same protein and affect that key shape. A couple of other key terms. We talk about the affinity of a drug. This is how strongly it is attracted to a receptor. And so a drug with a high affinity, even in very low concentrations, will still bind to the receptor. And a drug with low affinity, you would need much, much higher concentrations to get any kind of binding to the receptor at all. And so a drug with no affinity does not bind to the receptor. So once again, just getting all of the terms distinguished one from the other, it's key to understand that agonists and antagonists can both have affinity. They both can bind to the receptor. But another term is efficacy. Efficacy, that intrinsic activity in the body, or another way of putting it, the intended effect. So if a drug binds to the receptor and has no effect, it has zero efficacy. So technically speaking, that's how we define antagonists, because they bind to the receptor, but you don't get the normal intended physiological effect at that receptor, so that's actually rated as a 0. So drugs that bind to the receptor and at higher concentrations have the same effect, that maximal response, as the endogenous substance. They're given an efficacy of 1. When we look at the graphs, we'll talk about that as a 100% maximal efficacy. So agonists have affinity because they bind, but furthermore, agonists have efficacy. A lot of prescribers challenge the use of efficacy only as the measure in randomized controlled clinical trials by which drugs are finally approved for the market. As we've seen, there's a lot more data than just efficacy alone. So it's important not to say that efficacy means exactly the same thing as effectiveness. Efficacy is, can the drug work? And effectiveness is, does the drug work? So the distinguishing feature here with effectiveness is it's in the clinical setting, with that real person and all of their genetics, good health and bad health, and we're just looking at where they as an individual would fit on that normal graph of the general population in which that drug was first tested. So that efficacy was a population measure, and effectiveness is just that individual. Does the drug work? So the same thing with saying that antagonists don't have efficacy, but they certainly can have effectiveness. So antagonist, remember, bound, doesn't have the intrinsic activity, doesn't produce that effect. But the intended result still happens. You intended that drug to do some blocking. This happens instead. It blocks that natural ligand from binding. So efficiency or effectiveness, better than just can the drug work, can the drug work as intended, might be, as proposed by some prescribers, a better distinction. But this efficacy is actually a mathematical concept, which we will look at when we actually measure the responses of drugs to their agonists. As you can see, when you apply a drug to either a living system, or perhaps you're doing tissue studies on, say, the smooth muscle in the airways of the lungs, and looking at concentration of an agonist drug so that you do get a response, and you plot that against the response in that tissue. Let's say it's this smooth muscle in the airways, and the smooth muscle is going to dilate in response to that drug. We just measure how much of a response you get. And so there's the 100% maximal response, and you can see that it takes high concentrations of this drug to produce that response, and eventually this rapid exponential growth might start to plateau out for this particular drug. Now, we're also interested in measuring this particular point here. It's still an effective concentration. It still produced an effect in the tissues, but it produced only 50% of that maximal response. That measure there, that drug concentration, is a measure of potency, how much that drug binds to the receptor. So remember that potency and efficacy in pharmacological terms were not defined the same. Potency just was more neutral. It implied just the substance binds, and measuring how much. And we said even antagonists will have affinity. But here, potency of an agonist can be measured by taking that 50% of maximal response, EC50, and now you can compare drugs to say which one binds stronger or weaker. Note once again that potency clinically is less important than perhaps that maximal efficacy response. Potency less important clinically than this term efficacy, and yes, challenged by some prescribers that more important should be placed on effectiveness, individual responses, but more on that later when we look at the drugs affecting each of the body systems. Just to get these key concepts, some of these are very necessary for you to get an appreciation of toxicokinetics later. So that EC50, concentration at which 50% of the maximal response was produced, that's a potency that we can now use as a comparison for other drugs. Now, because the responses to drugs get exponentially rapid, we're most interested in studying that part of the curve where you're starting to get close to the maximal response. In researching these drugs, pharmacologists do a mathematical calculation called a semi-logarithm. And when you do that calculation, it's done actually on the graph paper itself. So instead of getting that rapidly increasing curve, it turns the drug dose response curve into this beautiful S-shaped curve, a sigmoid shape. And the key thing here with this mathematical calculation is that effective concentrations between about, as you can see there, 20% and 80% come out on this particular sigmoid graph as a straight section. And you can still measure that maximal efficacy level, the 100% response, and so that's called EC100, effective concentration 100%. Most drugs are given at an EC80. So now analysing that term, that would be an effective concentration 80%. The drug is not given so as to give the maximal response in the tissues. 80% is sufficient and will also perhaps reduce the risk of adverse effects. At that EC100, there would be a greater chance of that drug showing adverse effects on other unwanted receptors or unwanted proteins in that body, in that person. So here's some graphs showing you that sigmoid curve, where once again we look at agonist drug concentrations on the horizontal axis. Once again, we're looking at a desired response, the normal physiological response, and let's keep using bronchodilation as our intended effect. So we're looking at agonists that would produce the same effect as the normal tissues. When you then plot three different drugs, A, B and C, you begin to see that drug A produces a much higher maximal effect, and drug B produces less of a maximal effect than drug A, and drug C produces much less maximum response, much less bronchodilation in this particular case. But when you look at that key straight part of the sigmoid curve and measure that EC50, all three drugs, A, B and C, have the same concentration at which you get a 50% response. And remember, that was that measure of potency. So they all seem to be as potent as each other, but certainly not as effective as each other. And so showing there efficacy, maximal intended response in a randomized controlled clinical trial ideal setting is certainly not the same thing as potency. Potency is more related to affinity, and efficacy is more related to clinical effectiveness. Though we said efficacy and effectiveness need to be distinguished as two separate terms, and that efficacy is in that ideal setting and effectiveness is, that individual sitting in front of you, how did they fit onto that curve of the population data as far as effectiveness of this drug. And there are more variables impinging on the effectiveness of the drug, whereas efficacy is an easy mathematical calculation. All other contributing variables have been somewhat ideally controlled in measuring efficacy. So, to look some more at antagonists, now that we've looked at agonists, where with agonists we use a dose-response curve measure. We'll see the same can be done with antagonists, because you don't get the intended response, though. You don't get a bronchodilation, which you can then compare to the normal endogenous ligand, that the way the dose-response curve is obtained is slightly different for an antagonist. So we said, technically speaking, that ideal mathematical measure of efficacy, antagonists don't produce that efficacy, though they can still have affinity, they still can bind. The key thing, though, was that the agonist stopped the endogenous substance, an endogenous agonist, from binding. Here's an example, but this particular example has sometimes actions in the bronchi on the smooth muscle. Sometimes these are intended, sometimes they're not. They are adverse effects, but these particular drugs, beta blockers, tend to be used more in cardiovascular indications, as we'll see later. But just to get you used to the terminology, they are beta blockers, they are antagonists. So there's a particular receptor in the body, in this case that beta adrenoreceptor or beta adrenergic receptor that you studied in physiology, and these drugs antagonize it. So a beta adrenoreceptor antagonist or a beta blocker, blocking the actions of the normal endogenous agonist. In this case we've used adrenaline as the example because it's one of the endogenous substances that can bind to these beta receptors. Now normally adrenaline would speed up the heart rate, so a beta blocker reduces the heart rate. So an antagonist can still have clinical effectiveness, though it has no efficacy, this ideal mathematical measurement of agonist compared to normal physiological substance. We did say that with antagonists there are those that compete, and we'll see that they are competing for an actual shape on the protein, lipoprotein or glycoprotein, and that competition could be reversible or irreversible. drugs that are non-competitive tend to bind irreversibly, but this is not always the case. With competitive reversible antagonists, they will interfere with the way the endogenous substance binds at a particular shape on the protein. So here we've shown the protein in purple and the agonist with a unique shape that then locks into that shape on the receptor. Our reversible antagonist might bind at the same site on the receptor as the normal agonist. The way to test that is to use concentrations of the blocker. If, as you increase the concentration of the antagonist, this leads to less and less of the agonist that can bind and have its effect, but all you have to do then is just raise the levels of the agonist, increase its concentration, and if that overcomes the antagonism, then those two chemicals, agonist and antagonist, were competing for the same site. If by increasing concentration of the agonist, it then wins out in the competition. You see, you have a competitive antagonist, a competitive blocker, blocking at the same binding site. So here is our semi-log paper measuring increasing concentrations of the drug and looking at maximal response in that tissue compared to the 100% maximal response given by the endogenous substance. And so here are some drugs. Drug A that gives the same physiological response as the endogenous substance. So it's an agonist. Remember, it's only with agonists that we do this S-shaped curve. But this is how we measure antagonists. We look at how the presence of that antagonist and at that particular concentration affects the normal sigmoid curve. And you'll see there that when you take drug A, the one that produces the same response as the endogenous substance, and put it with a competitive antagonist, drug B, the effect is to shift the dose-response curve to the right as the reader looks at the sigmoid curve. So another way of saying that, shift to the right. You can see that by having an antagonist in that system, let's say it's some smooth muscle airways tissue from the bronchi in the lungs, we're using a drug that has the same bronchodilatory effect as the endogenous substance, adrenaline, and when you have an antagonist, the S-shaped curve was moved to the right or away from the vertical axis, or it was moved up the number line of the horizontal axis drug concentration. Now, irreversible antagonists bind. They may bind at a completely different site to where the endogenous substance actually does its binding. We'll see that that can totally change the shape of proteins, and so the endogenous agonist, the chemical, in this case adrenaline, that would normally bind to those receptors in the lungs and on that smooth airway tissue in the bronchi and bronchioles of the lungs, can no longer bind. That target is unavailable now because this antagonist has bound. But if it was an irreversible antagonist, the action is quite prolonged. It doesn't terminate until you've removed all of those receptors and replaced them with new ones. We saw how we were going to look at that effect of irreversible antagonists. We used aspirin binding to its cyclooxygenase enzyme in platelets as an example, and how the action of aspirin on those platelets isn't terminated until those platelets are terminated by your reticuloendothelial system in a variety of organs of the body, particularly the liver. And you will, in the bone marrow, make new platelets, and then and only then do you terminate the action. Those are new receptors unaffected by that drug, aspirin. So how long an irreversible antagonist works actually depends upon body responses and on that receptor turnover rate. The way we then mathematically measure the actions of antagonists will differ. So reversible antagonists, we can mathematically measure them, even though they can't produce a dose-response curve, but we can mathematically measure reversible antagonists by whether they shift that normal dose-response curve for the agonist substance. And with irreversible antagonists, we have to look at other physiological and clinically relevant actions in the body. So all of these drugs, whether they are agonists, competitive antagonists, or these irreversible antagonists, all of their pharmacological effects and therefore their duration of action in the body are heavily influenced by those pharmacokinetic parameters. So I'm hoping that you're getting very used to those 4 key processes: absorption, distribution, metabolism, and excretion, and getting used to being able to describe them as pharmacokinetic processes measurable by pharmacokinetic parameters. So here's a non-competitive antagonist. Here we've shown a protein. In this case we picked an ion channel. It might not necessarily be a transporter channel. It might be one of those ligand-gated ion channels. So there's a particular site where an agonist drug can bind to activate that channel, produce an action in the body. Here we have a functional antagonist that binds somewhere else other than the site where the agonist binds, but by binding it can completely block that channel to the normal action. Those ions that would normally be moved through that channel either into the cell or perhaps even out of the cell, that action no longer happens. So when it's competitive, the binding tends to be more electrostatic. It's more about the pH, the salt bridges, little cation and anion bonds that can form between that particular endogenous compound and the receptor, or the agonist chemical and its chemical structure and the receptor. But if it's a non-competitive antagonist, it bound at a site other than the receptor site. It still inhibited the response by the endogenous substance, and the site could no longer function normally. In this case, the ion channel, which would normally be opened by the stimulation of the receptor, would stay in the shut position due to the action of the non-competitive antagonist. And as a general rule, non-competitive antagonists don't just bind electrostatically, they can also have some covalent binding to that particular protein.